Hi, my name is Mike, and I'm going to present my Exam 1 presentation. Of the two slides I've chosen to present today, the first one is the life cycle of moss, and the second one will be about the structures and the differences of monocot and dicot stems. So yeah, let's get into it. So in this first presentation, this is the life cycle of a moss. And in the life cycle, there are 7 stages, and each stage I will explain. So in the first stage, we have a sporangium, which houses spores, and it disperses its spores via wind transfer. Um, in the wind, once it transports itself, it lands on moist ground where the spores begin to germinate and bud, and that happens in stage 2. At stage 2, the haploid species begins to develop into one of two genders of gametophytes. Uh, we have a male gametophyte, which develops male gametes or sperm, which is called antheridia, where in the female gametophyte species, we have them develop female gametes or archegonia, which has eggs. At stage 4, we can see that the sexual process begins where by moisture, sperm is transferred towards an egg and begins to swim down into its core to develop into the new diploid species known as the zygote sporophyte. Um, at stage 5, it begins its development while housed inside of a gametophyte. And the sporophyte is then not only housed, but also fed and begins developing with the aid of the gametophyte. At stage 6, you can see that the sporophyte has now sprouted, and with the help of the gametophyte, is still housed within it. And the gametophyte begins to elongate its structure as well, keeping up with the length of the sporophyte until it splits at the middle. And at stage 7, we can now see that the sporophyte has developed a capsule-like structure on its tip known as the calyptra. And that is pretty much the structure that houses the sporangium, which breaks out at stage 1, which is what we see now. And that sporangium, that newly developed sporangium, will begin to disperse its own spores and repeat the cycle all over again. So that is pretty much the life cycle of moss. So if we go to the second picture, we can see at this stage right here, we can probably say that this is either stage 7 or 1, the beginning and the end of the life cycle, where we can clearly see sporangia within the moss itself, and the calyptra has been split already, so it's probably looking forward to dispersing its spores. So that is my first presentation in the first slide. So now with the second one, we have the tissues in dicot and monocot stems. So the difference in structure between dicot and monocot is that it is complexity. So in dicot, we have many layers in its structure versus a monocot stem, which looks more simple, but certain structures within both do share the same structure, or not structure, function. So beginning with the dicot, we have at the innermost layer, we have what is known as the pith. The pith layer houses ground tissue. And in this ground tissue are cells with many spaces of air in between, which help with gaseous exchange, as well as housing water, as well as transporting other nutrients throughout the entire plant. And with that, we have what's next is an arranged ring of vascular bundles. And these vascular bundles are transporting bundles, which help aid in nutrient, water transport as well throughout the plant, but also aids in secondary growth, which helps to widen the girth of the plant as it develops. So there's that. Within each vascular bundle, you can see that there is a xylem and a phloem, which help aid in that secondary growth that I mentioned before. Um, Outside of that, we have a very thin, almost outer layer known as the cortex, which acts as like a permeable layer uh, to help with moisture coming in towards the plant and then into its innermost layer known as the pith. and also other nutrient transfers, which help to diffuse itself within it. And on its outermost layer, we have what is known as the epidermis. And in the epidermis, it acts more of as a protective layer against pathogens, against harmful rays from the sun, and other predatory creatures that might prey on um, these kinds of plants. So that's what that does. Now, if we move towards the monocot, in the monocot, we can just tell just by looking at it, it's not as layered or if at all compared to the dicot stem. Uh, at the center of the monocot stem, we have ground tissue, but is not called a pith because in the dicot that is the layer within the structure, and that is what it's called. It's called the pith. But because there is no layer within the monocot, it's just ground tissue, but still has the same cells and spaces in between to support gas exchange, water storage, and other types of transfer of nutrients throughout the plant. And if we can see within the ground tissue, we see tiny, tiny vascular bundles, which also have phloem and xylem in them, and a cambium that cuts through it. That's what divides them into those two forms. And those help, aid in secondary growth as well as transport of other nutrients and such within the stem. Outside of that is just another epidermis, and that shares the same function as the epidermis in the dicot, where it just protects things, protects from things like the sun's harmful rays, pathogens, other predatory creatures. And yeah, so in that, we can see that its structure is very different in its complexity. Its arrangement of the vascular bundles are very different, where we can see dicot is much more organized and arranged in a ring, whereas in the monocot, they're more less organized, definitely towards the outer edges of the stem itself, but are scattered throughout the ground tissue as well. Um, and we can tell just by looking at the labels here within the dicot, there are several more structures to a dicot versus a monocot, which is more of like a foundational groundwork, basic cable representation of what that plant looks like. So, yeah, that is my presentation in total. So we've discussed the life of moss, its stages, uh, and now we've also discussed the tissues within dicot and monocot stems. And that wraps up my presentation. Thank you so much for letting me share with you.